All right, guys, we're going to start our unit on viruses. First things first, essential question is what are viruses and how are they different from living organisms? And so we're going to go through all of that stuff. Go ahead, and there's no note set up. You're just going to write down what I highlight and what I let you know to write down. Okay. So, first question is, are viruses living or are they non-living? Choose green here. Are viruses living or non-living? Write that down. Viruses are considered non-living. In other words, they are abiotic, and they have several reasons why. They do have some properties of life, not others. Viruses can be killed. Okay, so that's a property of life. They also have a protein coat, so they are made of protein, and they also have genetic material. So those are things that kind of give them the impression that they are alive, but unfortunately they cannot maintain a constant internal state or homeostasis. So they don't have metabolism. They cannot control their temperature or anything of that nature like we can. In addition to that, they also cannot reproduce without a host cell. So they can't reproduce on their own. And so that also goes against those characteristics of life that we talked about at the beginning of the school year, whether or not an organism is living. And so one of them was the ability to reproduce. Another one was the having of cells, and viruses don't have cells. Another one was the ability to maintain a constant internal state or homeostasis. And the other was having a metabolism, so the ability to convert energy, whether that's converting solar energy into chemical energy or chemical energy into cellular energy like respiration. So they can't do any of those things. So we consider them non-living. Okay, so let's go ahead and answer this question. What are viruses? A virus is a non-cellular particle, and when I say particle, I mean it is extremely small. It does have genetic material on the inside and a protein coat on the other side, on the outside. And what it does is it invades living cells. Okay, so viruses are non-cellular particles. They have genetic material on the insides, and on the outside there they have a protein capsid or protein coat. So this outside right here is made of protein. And they come in all sorts of various shapes, but the majority of them are extremely tiny. And in fact, even the largest one, which I think is Ebola, is actually not that big either. So they're very tiny, 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 much smaller than the smallest cell, the smallest bacteria cell. Viruses are very tiny. So you're not going to write down any of this viral history. I'm just going to go over a few things. So the individual that is credited with developing the first vaccine is Edward Jenner. So he developed a vaccine to prevent smallpox, and he started with the cowpox virus. Okay, so his invention has allowed us to eradicate smallpox and also allowed us to create new vaccines for other viruses. So Edward Jenner is credited for that. Like I said, viruses are extremely tiny. So they're smaller than the smallest cell. They're measured in nanometers, and so that is 1 times 10 to the -9. That's pretty small. Okay, you cannot see viruses unless you use what's called an electron microscope. Okay, so electron microscope. Electron microscopes are extremely expensive, and usually there's only a few on like a university campus. So they're not something that we'd have here at school. So essentially, they would be used to pick up these small, very small viruses, these very small particles. Okay. All right, so let's look at the sizes of viruses and just kind of get an idea in our minds, the comparison between the size of a virus and that of a cell. So if we're starting off with the human red blood cell, at the diameter is 10,000 nanometers in length. And so this is what it looks like right here. Okay, so that's just part of it. So it's, you know, relatively big. Okay, and then you go down to the bacteria cell, E. coli. Okay, and it's 3,000 by 1,000 nanometers. So it's actually way smaller than the human red blood cell, but you guys knew that bacteria are smaller. But let's look at the viruses. The largest virus, the Ebola virus, 970 nanometers. And that's as big as it gets. It doesn't really get much bigger than that. And so going through some of these other viruses, let's look at the polio. Here's polio. Polio is pretty tiny, 30 nanometers. Like that's really, really, really tiny. The rhinovirus, which is the cold virus, adenovirus, those are cold viruses too. They're all super tiny as well. They're so small. And this is literally, if you look at this this diagram, this is a comparison of how small the virus is compared to how large a cell is. So they're very, very tiny particles. All right, so let's talk about the characteristics of viruses. Generally speaking, they have a protein coat called a capsid, and they have a nucleic acid core that has either DNA or RNA. And they're only capable of reproducing in a host cell. So they cannot reproduce by themselves. Okay, obviously, as I said before, they are non-cellular. They are not made of cells. Okay, so these are your basic characteristics of viruses. Okay, they're not living. They don't have cells. They're a protein coat with DNA or RNA inside of it. And the only way that they can reproduce is by hijacking a host cell. Okay, go ahead and draw and label this diagram. Draw and label this diagram. To the best of your ability. I know some of us are really good at drawing and some of us aren't, and that's okay. But if we're looking on the outside, you've got these spikes here. All these spikes are for attaching. Okay, for attaching. Viruses are host cell specific. So essentially, viruses won't just attach to any cell. Like the COVID virus only attaches to cells in the respiratory system. It doesn't necessarily attach to cells in the digestive system. So these are specific. So they attach to specific cells. Then this particular virus has an envelope. Not all viruses have an envelope. It's like an extra layer of protection. And then, of course, there's the protein capsid, which is yet another layer of protection. So this particular virus has two. And then so you've got DNA on the inside of this particular virus, but bear in mind it could also be RNA. It just depends on the virus. Okay, so have spikes to help attach to hosts, and viruses infect specific host cells. So write that down. Okay, so a few more characteristics. You've got these protein subunits called capsomeres, and not all viruses have them, but essentially this is where you would find them on the outside of the virus. So outside of the host cell, the virus is inactive. It can't do anything. Okay, it doesn't have ribosomes. It doesn't have organelles. It doesn't have cells, and so it lacks ribosomes. Okay, and it uses the raw materials and enzymes of the host to reproduce. But if it's outside of the host, it can't do any of those things because, well, it lacks what it needs to do those things. Okay, here is an electron microscope picture of Ebola right here. Okay, it actually, to me, kind of looks like a worm, but it's not. It's incredibly smaller. And then here's HIV. Okay, viruses can cause disease, and so examples of viral infections would include anything that ends in pox. Okay, measles, the flu, colds, warts, AIDS, Ebola, COVID. Go ahead and add that as your example. Herpes, which, you know, really, truth is, anything that ends in pox, as well as measles, those are all belonging to the herpes family. So those are herpes anyway. And some viruses can cause cancer. So the human papillomavirus causes cervical cancer. Okay. So those are some examples of viruses that cause disease. All right, so various shapes, and you just saw that in the diagram that I showed you earlier. Ebola is kind of helical, and then the flu is kind of like a polyhedral shape. And then bacteriophages look really weird, and so I'll show you that in just a moment. Okay, so there's Ebola. Here's an adenovirus. Okay, covirus. And this is the bacteriophage. And so to me, bacteriophages kind of look like little robots, like really look like little robots. Go ahead and draw this. Label the capsid. So this is a capsid. All of this is your capsid. Label the DNA. Okay, so all this stuff on the inside is the DNA. Label the sheath. Or the tail fiber. This is the tail fiber. And then here's an actual picture of what those look like. Now, bacteriophage only, you can write this down, only attacks bacteria. So only attacks bacteria. Okay. So here's a picture, really gross picture of herpes. This would be simplex 1, and simplex 2 would be someplace else. Here's the adenovirus, which is the cold virus. Looks very similar to COVID. Here's the flu virus. Here's chickenpox. Here's the human papillomavirus. causes genital warts, and if it gets bad enough, can cause cervical cancer. All right, so how do we identify viruses? So identification. A virus is either DNA or RNA, so it has DNA or has RNA. It may or may not have an envelope. We might look at the shape of the capsid. We also want to know the host they infect. Now, some viruses are going to infect multiple hosts, so a virus can jump from bat to human or mouse to human. All right, so again, viruses that attack bacteria are called bacteriophage. And so this virus would attach to the cell, inject its DNA. That DNA would become incorporated with the bacterial DNA, and then it would take over, and it would force the cell to begin building more viruses, and all of those viruses would accumulate in the bacteria cell, and the virus then explode out of the cell and lyse the cell, kill the cell. Okay, so there is another example or another diagram of the bacteriophage. Okay, moving on to retroviruses. Go ahead and title this retroviruses. Retro means like going backwards. And retroviruses kind of do go backwards. So let me explain that. All right, so retroviruses contain RNA and not DNA. So when it infects a cell, it injects its RNA. The issue with RNA viruses is that they mutate way more often than DNA viruses. So you end up with all these different variants. So COVID, influenza, all these HIV are all RNA viruses or retroviruses. And because there's so many different variants of the virus, it can be very difficult to vaccinate against it. So because this virus has RNA and not DNA, it has to do things backwards. So kind of going back down memory lane. Previously, we talked about protein synthesis, and we talked about how DNA is transcribed into mRNA. Well, if there's only mRNA, what needs to happen is that mRNA has to get reverse transcribed into DNA and then transcribed again into RNA. Okay, so we start with RNA and we go backwards. Okay. I know this is going to be kind of hard to draw. Let me see if I can find a better one. No, that's just as bad. Go ahead and draw this diagram. So these little spikes right here, the glycoproteins, therefore attaching. Okay, attaching to cell. So if this is HIV, this virus would attach to white blood cells because HIV attacks the white blood cells. Okay, and then we've got the envelope, which not all viruses have. And there's the capsid right there, made of protein. In the middle is that little strip of RNA, viral RNA. Right here, you've got your reverse transcriptase. Okay, draw that and label it. So examples of retroviruses are HIV and AIDS. All right, so time to start doing a whole lot more drawing. All right, so we're going to start with, go ahead and label this part, viral replication. Like I said, viruses are very specific in the species they attack. And they're also very specific to what cells in that species that it attacks. Sometimes humans do share some diseases with animals. Okay, so like the swine flu, bird flu, rabies. So going through the lytic cycle, label this lytic cycle. Let's go through the steps. Step 1 is attachment to the cell. So the virus attaches to the cell. Step 2 sounds real funny, but it's penetration, where the virus injects its DNA into the cell. Then replication, where the viral DNA is replicated and then it is translated into proteins. And then assembly, when the pieces of the new viruses are put together. And then the end is the release. That's when those new viruses now have no more room in the cell, so they have to burst out of the cell. And that kills the cell. That's what cell lysis means. It means the cell is broken. It has burst. Now all these viruses have burst out of the cell. Now they're going to go and attach to other cells. I'm going to start this whole process all over again, so this process just repeats itself. It's going to be very hard, but I want you to draw this. So here's your step 1. You can pause. There's your step 2. Here's step 3 and 4. And then there's step 5. So let's go through the steps again. Here's step 1. So pause right there. Attachment. Here's step 2, penetration. Pause right there and draw it. Step 3, DNA is copied. Step 4, assembly, so putting all the viruses together. And step 5, they lyse the cell and get released and start all over again with a new cell. Okay, so I should see 4 pictures, actually 5 pictures. Okay. Pause as many times as you need to. You've got 5 pictures to draw there. So attachment, then penetration here, then DNA gets copied, proteins get made, and the virus is assembling, and it lyses the cell, destroying the cell. Now, that's the lytic cycle. The other type of cycle, in my opinion, is much worse. It's called the lysogenic cycle. Okay, so lysogenic is slightly different. The virus has the ability to become dormant inside the cell. So let me explain how that works. So that's called latent viruses. They can remain inactive for a very long period of time, sometimes years. So chickenpox is one of these lysogenic cycles. What happens is that you get over the chickenpox, but the viral DNA stays in your DNA. And then when you get older, it kind of wakes up and it goes into the lytic cycle, and that becomes shingles. Okay, so later they activate and produce new viruses. Now that could be really, really awful. My understanding is that shingles is horrible and painful and nobody wants it. So fortunately for you guys have already had a vaccination to the chickenpox. Lucky you. But when I was coming up, that did not exist. So we had to actually catch the chickenpox. So examples are HIV and herpes. Those are latent viruses. HIV, you can be infected with HIV and not know it for years, and then it wakes up. Herpes, same thing. That's how people get a cold sore, every month or every year or something. It's because the herpes viral DNA is just hanging out in their DNA, and it just wakes up randomly and decides to go into the lytic cycle. Okay, so we're drawing again. There's your step 1. And here's your step 2, draw that, penetration. And then something different happens here. Your step 3 is that this viral DNA gets incorporated into the bacterial DNA. And it could be there for however long. Then one day it just wakes up. And step 4 is when the virus replicates Step 5 being when the virus is assembled. And then step 6 is the lysing of the cell and the virus is released. So go back and play that over and over again. You should have 6 steps. The lysogenic cycle. Make sure you title it lysogenic cycle. So there are 6 steps. So it's just like the last one. The only difference is that instead of immediately forcing the cell to replicate its DNA and build viruses and the viruses burst out of the cell, the difference is that the viral DNA gets incorporated into the cellular DNA and it sits there for a while, maybe a couple of days, a couple of weeks, could be years, could be decades. And then one day it wakes up and it goes through the lytic cycle. And so there's that one extra step, which is a doozy, where it just kind of sits there in the host DNA. Okay, so same process. Okay. Okay, so this is the lysogenic cycle. Virus attaches, injects its DNA. DNA gets copied. Viruses get assembled. Viruses are released. And then, of course, it happens all over again to a new cell. That's the lytic cycle. Lysogenic is different in the sense that this virus attaches, injects its DNA, but instead of copying that DNA and letting it just, instead of copying that DNA and then forcing the cell to make more viruses, instead, it's going to copy the DNA, but it's going to incorporate that viral DNA with the cell DNA. And then the cell is going to continuously divide as it would normally and continuously copy that viral DNA. So after a while, you end up with a whole bunch of cells with the viral DNA, not just one here and there. Almost all the cells have the viral DNA because, well, it's been copied so many times. And then one day it might decide to wake up and start assembly, start to copy the DNA and force the cell to assemble viruses. And that's the lysogenic. This is the one that's kind of sneaky. So again, your example is the chickenpox. Can reappear later as the shingles. Not pretty. I've heard that is extremely painful. Like having cold sores on your back. Herpes, all of the herpes families, so anything that ends in pox is a herpes. Obviously, herpes simplex 1, simplex 2, measles is a herpes, cold sores are herpes. Okay, herpes can last an entire lifetime. So genital herpes or cold sores, which are simplex 1. So that is a forever thing. All right, so really there's very few treatments for viral infections. So when you get the flu or if you got COVID or what have you, typically you just have to wait for your body to fight it off, your immune system. And usually what happens is that you've got these very large cells called macrophages. And macrophages, what they do is they go along and they look for infected cells. So cells where the virus has hijacked the cell and is forcing the cell to make viruses. And before that cell explodes, the macrophage will gobble it up and destroy everything that's inside of it. And so, obviously you've lost your cell, but you're going to lose that cell anyways. So your immune system kicks in once you are infected with the virus. But on the other hand, you can prevent infection by getting a vaccine, which is a weakened virus. Some of them, like the COVID vaccine, is not a weakened virus. It's actually very different. It's a piece of mRNA. And so it's actually kind of revolutionary. But generally speaking, vaccines when you get a vaccine, you get a small amount or a weakened version of the virus itself. Okay. And the idea is to stimulate an immune response, but not cause illness. So you want to create that immunity, because once you stimulate an immune response to that virus, your cells and your immune system are always going to remember it. So if it ever sees it again, boom, it's there. So you kind of introduce this virus to your immune system, and your immune system will always, always, always defend you against it. So that's what's kind of nice about getting a vaccine. All right, so that's everything about viruses. Please write all of that down.